excited to present our speaker today, who's a PhD student in my lab. She's been in the lab since January 2016, and at the timing of all this was set to start her very last experiment for the last chapter of her thesis, which I'm sure will get done at some point. But up until now, she's got a lot of great data to show you. And she's really spearheaded a lot of work in the lab related to longitudinal animal imaging, kind of from a technical standpoint, how to deal with treatment, how to deal with some imaging at some animals at some younger time points. So it's really exciting to see it all come together kind of in this examination of risk factors for neuropsychiatric and neurodevelopmental illnesses. So Lisa's a really well-rounded student. She does a lot of work in science communication. And before she came to us, she did a master's with Bruno Giros, also at the Douglas, starting in 2013, and her undergrad in neuroscience and in Carlisle, Pennsylvania, at Dickinson College. So thanks a lot for still doing this talk, Elisa, and thanks for coming here. And just a reminder to everybody, this session is being recorded. So if you want to ask questions, feel free. You can raise your hand using the raise hand button next to, I think it's somewhere. You can't find it. If not, you can chat me directly, and you'll be welcome to ask questions then. If you can mute yourself as well, that'd be very much appreciated. Thanks. So Lisa, I'll let you take it away.
Great. Thanks so much for the introduction, Mallar, and thank you all for being here. I'm really excited to share with you my work on maternal immune activation and how that affects offspring brain development. So just jumping right in. So we know from epidemiological evidence that maternal infection has been implicated as a risk factor for autism spectrum disorder and schizophrenia, amongst other disorders. And from the schematic here, we have kind of the current model of how we think that MIA leads to psychiatric disorders in offspring. So we could see that an infection in the mother leads to a release of pro-inflammatory cytokines in the bloodstream, and these are thought to alter the brain development of the offspring. In addition, a combination of the genetic background or autoimmune status of the mother, as well as potential second hits experienced during childhood or adolescence, such as stress, neglect, abuse, or exposure to drugs of abuse, combine with the consequences of maternal infection to increase the likelihood of offspring developing psychiatric disorders in adulthood. And here we have data from a birth cohort study in which serologic evidence for exposure to prenatal influenza specifically was recorded, and offspring were followed up for almost 40 years to determine whether there was an emergence of psychotic disorders. And what they found is that the risk for schizophrenia was increased sevenfold for exposure in the first trimester, and threefold if exposure occurred in the first half of pregnancy. And similar associations have also been made for other neurodevelopmental disorders such as autism, as well as other infectious diseases such as rubella, measles, herpes, and more, which suggests that the neurodevelopmental insults are not due to specific pathogens, but rather the inflammatory response of the mother. And while this work is extremely important and very exciting, it also has its challenges, as it takes almost 40 years to accumulate these data, which is why we can turn to animal models as useful tools of investigation, which is what I've done in my work. And another important consideration is that not all gestational stages are equal. Neurodevelopment in the first trimester differs from that of the second and third, as you can see here in this schematic. So you might imagine that a disruptive factor like MIA would affect something like neurogenesis or gliogenesis if it occurs in the first trimester, whereas it might impact more of a neuronal migration if it occurs in the second trimester, or myelination in the third. Interestingly, there's been some attempt to understand whether a certain trimester or gestational window confers exposure during certain windows confers greater risk for schizophrenia or autism onset later in life. But there's no like super clear evidence, although the first and second trimesters have been more implicated than the third. And so, as I mentioned, animal models are extremely useful tools for investigating these types of questions. And prenatal MIA has been modeled in rodents for the past few decades using either bacterial mimetics or viral mimetics. And in my case, this is what I've used, poly-IC. It's a double-stranded analog of RNA. So this is a byproduct of viral replication. And when it's injected into the pregnant mother, it causes an inflammatory response whereby you get an increase of pro-inflammatory cytokines in the maternal compartment. These can then cross to the placenta where they can increase inflammation locally, and some can actually cross through the placenta and reach the fetal compartment where they can then either activate the fetal immune system and cause developmental and structural disturbances in the fetus. Okay. And so last year I spent quite a bit of time reviewing the literature on studies looking at MIA. Sorry, it's a bit cut off. It's better. Looking at studies that use magnetic resonance imaging to investigate the effects of MIA on offspring brain development. And I've summarized them in this figure based on 5 species: the human, the mouse, the rat, the rabbit, and the macaque. But this is a really busy slide, so for today we'll just focus on we'll focus on the human and the mouse. So as you can see, the timeline starts at conception, and then we have the 3 trimesters highlighted here with important neural in blue and immune in purple events kind of highlighted throughout the developmental time window. Then we have birth and the postnatal period. And so what you could see highlighted are the different timings prenatally at which MIA was either observed or induced, and then postnatally the timings at which the offspring were evaluated. And essentially the main thing that I wanted to draw your attention to is that there are very few studies actually comparing the gestational timing of MIA within the same study. Very few studies use females as well as males in their investigation, and this is particularly true for the preclinical work. There were very few studies in the rodent literature looking at MIA early in the lifespan, and most importantly, there were very few longitudinal studies leveraging MRI as, it's perfectly applied to be able to do longitudinal in vivo assessments. And the reason why I'm stressing this so much is that a lot of our understanding of human neurodevelopmental disorders actually has come from longitudinal mapping of developmental trajectories. So here we have a well-known model for cortical thickness development with age on the x-axis and thickness on the y-axis. And we have 3 different hypothetical curves for pathology in red relative to the healthy control curves in blue. And we could see hypothetical deviations in development, such as a delay in the peak thickness attained, a more accelerated decline over age, or just a completely flat curve, so no peak attained at all. And this level of characterization, as I mentioned, has been critical to our understanding of human neurodevelopmental disorders and should be translated back to animal work aimed at understanding risk factors for neurodevelopmental disorders, but this has not been done by very many yet. So this brings me to the goals of my work. We wanted to know how exposure in the first and second trimester to prenatal maternal immune activation might alter the development of the offspring brain and behavior. And the reason why I've quoted first and second trimester is because we're talking about the human equivalents here, but I'll explain that a little bit more as we move through the presentation. And we chose to look throughout the lifespan, first starting from childhood to adulthood, and then we went back into the fetal and neonatal developmental windows as well. So to address this first question, we injected pregnant dams, so these are C57 black 6 mice with either poly-IC or vehicle control at gestational day 9 or 17. And then we imaged the offspring throughout development, starting at weaning, then adolescence, early adulthood, and later adulthood, using structural in vivo MRI. And just to reorient you back to the human equivalents, GD9 corresponds to the end of the first trimester of human gestation, and GD17 to the end of the second. And mice are actually born at the, I guess, equivalent of the beginning of the third trimester in human development. So that all occurs postnatally for them. And by the end of their first week of life, they're more equivalent to a human at birth. So I guess these differences in development should be taken into account when performing these studies. So in addition to the structural MRI, we also performed a series of behavioral tasks that assay behaviors that are relevant to neurodevelopmental disorders, but I'll go into these a little more detail afterwards. And we acquired these behavioral metrics both in adolescence and adulthood. And in terms of numbers, we ensured to have both males and females for each group, as well as offspring coming from at least 5 different litters to account for potential litter variability in our study. So all of our imaging was performed at the Douglas 7 Tesla animal scanner. We acquired T1 weighted structural MRI at 100 micron isotropic resolution. And we used manganese chloride to enhance our contrast. And that improves contrast in areas like the olfactory bulb, the hippocampus, and the cerebellum, which then improves our subsequent image processing after the fact. Okay. And so to analyze our data, we use deformation-based morphometry to assess voxel-wise volume changes. And here we have an example cartoon of mouse brain 1 and mouse brain 2, and we could see that mouse brain 1 has a larger cortex than mouse brain 2. So we're gonna need to shrink this cortex and expand this cortex to align the scans into an average. And the degree to which we had to shrink or expand each individual voxels can then be backpropagated onto the original scans and give us a measure of local volume change at every voxel. And so this workflow was originally designed for cross-sectional data, but in our case, you might imagine that mouse brain 1 is actually the brain of one mouse at time point 1, and that the mouse brain 2 is that same mouse scanned again at a later time point. And in order to more appropriately model this longitudinal aspect of our data, we used a 2-level modeling Pydpiper toolkit developed by Jason Lurch's team. And simply, this works by creating an average of all the time points per subject to make a subject average. And then this subject average is all the subject averages are registered together to create a population average, which allows for a common space in which to perform our statistics. And for our statistical analyses, we performed linear mixed effects models at every voxel in the brain for the deformation fields at the subject level. And we use curvilinear fits to be able to compare slopes or trajectories, and corrected for multiple comparisons using the false discovery rate correction. Okay. So we first compared our 2 saline control groups and found that they weren't different. So we collapsed them into a single group and then compared our 2 poly-IC groups to the one saline control group. And here we have coronal sections of the mouse brain going from posterior to the anterior axis, as you could see by this little sagittal view. And we're looking at the significance map of differences in curvilinear age interaction with group, focusing on the difference between the offspring exposed to poly-IC early in gestation, so at GD9, relative to the saline control group. And what you could see is that there are significant and widespread differences throughout the brain, significant at 1% FDR, but thresholded at 5, just for visualization. And you can see the positive T statistics are in warm colors and negative in cool colors. So what we could then do is actually look at some regions of interest and plot the volume of peak voxels over age, so that we can actually see what the trajectories look like. So here we're looking at, sorry, where's my cursor? Okay, we're looking at age on the x-axis and a measure of proportional volume change on the y-axis. So everything at 1 is no different than the subject average. Everything above 1 is going to be larger, and everything below is smaller. So at 1.25, you have a 25% larger volume relative to the subject average. And I've chosen to focus on these regions as they're implicated in schizophrenia amongst other mental illnesses. So first we have the striatum, and what I'd like to draw your attention to is the pink curve for the early poly-IC group. And so as you can see, it starts smaller at weaning, P21, and then tends to overshoot and be larger in this adolescent early adult period, and then normalize in adulthood relative to the blue curve for the saline and the purple for the late poly-IC group. We see a similar curve for the septal nucleus, which has projections to the hippocampus and is also involved in memory, as well as in the CA1 dentate gyrus region of the dorsal hippocampus, although the overshoot seems to be slightly less pronounced in this curve. And finally, we also see it in the cerebellum, which is one of the later developing structures and perhaps more sensitive to neurodevelopmental insults. Okay, so next we can compare the curvilinear age by group interaction for the late poly-IC group relative to saline. And what we could see here is that the differences are more localized than the ones we saw in the early poly-IC maps. And they're also less statistically significant, only reaching a 10% FDR threshold. And again, if we go and look at some peak voxels, what we could see here is that the late poly-IC curve in purple seems to have a more is flatter than the other two curves in the left nucleus accumbens, for example. And in the left amygdala, again, we see kind of a flatter curve in the first stage of life, and then kind of more of a decline into adulthood. Whereas the other two curves follow a different trajectory.
Can I ask a quick question? Sorry.
Sure.
Oh, so how do you deal with head size, or like, do you need to deal with head size in these analyses?
So there are two different Jacobian determinants, I guess, that we can get out of our deformation analyses. So in this case, I'm modeling the absolute, which doesn't, I guess, correct for total brain volume, because we're interested in looking at the trajectory over time, but we can also use the relative determinants, which only look at the nonlinear deformation, so it would account for differences in head size, and we can look at more local changes that way. I don't know if that answers your question. So
yep, absolutely.
Okay,
thanks.
great. Okay, so finally we can compare the two poly-IC groups directly to each other. And what I'd like you to notice here is essentially these maps look very similar to the ones in which we were comparing the early poly-IC group to the saline. So again, we can see pretty widespread differences throughout the brain, again, significant at 1% FDR. And if we look at some key regions of interest, in the dentate gyrus, again, we see that the pink curve for the early poly-IC group follows that same undershoot followed by overshoot, whereas the late poly-IC group is pretty flat here in purple. Whereas in the cingulate cortex, we actually see that the early poly-IC group starts larger and then has a more accelerated decline over age, whereas again, the late poly-IC group seems to be a bit flatter in terms of their trajectory. And so based on these plots so far, it's quite clear that the exposure at the two different gestational timings causes quite stark differences in development for the offspring, and that the early poly-IC group seems to be the more affected one.
Elisa, there's just a question from Dominic Walker.
Yeah.
And she has a question about hemispheric side, lateralization. Is there a difference between left and right? Data is always shown for the left side, maybe by choice? Thanks.
Yeah, so there does seem to be some lateralization. What often happens is, I guess I did focus on the left a lot. What often happens is if I drop the threshold, then the bilateral side will come up. So I guess if you see in the left nucleus accumbens here, the right is also significant. It just like maybe not as much. But I guess I haven't taken as close of a look at lateralization. But yeah, even in the striatum, we do have effects on the other side. So perhaps if I shift, if I go a bit more posterior, the effect on the right side would be a bit more striking. But that's a good point. I think it would be useful to show not only the left side effects. Okay, so as I mentioned, we also performed a series of behavioral tests. So the first one I'll talk about is the open field test. And we use this as a proxy for exploratory and anxiety-like behaviors, both in adolescence and adulthood. So essentially what we're measuring is the distance traveled or time spent in the anxiogenic center zone versus the edges or corners of the arena. So what we could see is that in adolescence, the early poly-IC group travels significantly less in the center zone relative to the salines, as do the late poly-IC group, suggesting that they have a more anxious phenotype at this stage. Whereas if we look in adulthood, we actually see a reversal of this phenotype, with the early poly-IC group traveling more in the center zone than the saline controls. And we don't see any difference in the late poly-IC group. And originally I was fairly puzzled by this, but we might interpret it as an increase in perhaps risk-taking behaviors or impulsivity. We also performed the marble burying task as a measure of stereotypic behaviors, and this is central to ASD pathology, but also important for schizophrenia. And briefly, how this works is that we place a mouse in a cage with a thick layer of bedding and 15 evenly spaced marbles, and measure how many marbles a mouse buries in the 30-minute period. So the more marbles buried, the more anxious or stereotypic behaviors they display. And so similar to what we observed in the open field task, we see that the early poly-IC offspring bury significantly more marbles in adolescence, with a trend level increase in the late poly-IC group as well. And if we look at this behavior in adulthood, we see kind of a normalization with a trend level increase in marbles buried for the early poly-IC group and no difference for the late. Moving on, we also measured sensory motor gating behavior using the pre-pulse inhibition to an acoustic startle response. And the reason why this behavior is quite interesting is that the same task can be performed in humans, and it's been shown that individuals with schizophrenia also have impairments in this behavior. So essentially, we're detecting one's ability to filter out irrelevant stimuli. And the way the task works is you present an acoustic stimulation and then measure the startle response to go with it, and then see whether the presentation of a small pre-post tone prior to the acoustic stimulation is sufficient to dampen the startle reaction. So in a normal individual, it would, but someone with impairments in sensory motor gating would startle just as much, even if the pre-pulse was there. And what we see in our mice is that the early poly-IC group are significantly impaired in this behavior. So there is no ability to inhibit their startle response. And this is true if we look at this plot to your right for increasing loudness of that pre-pulse tone as well. And if we look in adulthood, this impairment seems to be normalized as well. So there are no differences between our groups in adulthood. And so essentially what we can think is that based on the behavioral data as well as the neuroanatomical findings, that many of the impairments that seem to emerge in the adolescent period tend to normalize by adulthood, which is quite interesting. So then, because we had a lot of behavioral data acquired for each mouse as well as the neuroanatomical data, we wanted to think of a way in which to kind of incorporate all of it into one analysis instead of looking at each metric alone. And we chose to use the partial least squares analysis in collaboration with Dr. Bratislav Misic at the MNI. And we used the data from the adolescent time point for this analysis, as this is where we saw the majority of our changes. So briefly, PLS is a multivariate method for relating two sets of variables to each other. And in this application, we used a brain variables, so our deformation-based morphometry metrics for our mice, as well as behavioral metrics, so data from all of the behavioral tests we used, we performed, as well as sex and litter size as kind of our demographics variables for the mice. So these are arranged in two different matrices with participants as the rows and the data as the columns. And first, the brain and behavior matrices are z-scored and then correlated, which gives us a correlation matrix for these two data sets. And this matrix is then subjected to singular value decomposition, which gives us a set of mutually orthogonal latent variables, which essentially can be thought of as patterns that describe the correlative relationship between the input brain and behavioral data. And so what you get out of a latent variable is a set of brain scores, which describe how each voxel weights into that given latent variable. Similarly, a set of behavior scores, which describe how each behavioral variable weights into a given latent variable, and then a singular value from which we can determine the proportion of variance explained by that latent variable. And we can then use permutation testing to evaluate the significance of each latent variable and bootstrapping to evaluate the reliability. And so in our analysis, we found two significant latent variables, but I'll only discuss the first, as the second one essentially was just the sex effect. So this first latent variable explains 36% of the covariance. And I know there's a lot going on here, but I'll try to break it down for you guys. So essentially in red, we see many cortical areas and striatal areas, and these seem to be associated with a pattern of increased anxiety based on the open field test, as well as less impairments in social behavior and sensory motor gating. Whereas for areas in blue, we can kind of flip this relationship. And so these tend to be more hippocampal, thalamic, and cerebellar regions, and they're associated with less anxiety, but greater impairments in social behavior and sensory motor gating. Okay. what we can then do is project the brain and behavior weights back onto the individual subjects and see how correlated they are with this pattern. And we can also color the subjects by treatment group to see if there are any clusters. And overall, I mean, there's no super clear pattern, but it's possible that some of the early poly-IC group in pink have a stronger correlation in this pattern. And then because we actually have longitudinal data on these mice, we can then take the weights for the brain and behavior scores calculated in adolescence and project it onto the adult data, so the raw brain and behavior data from adulthood, and see how those patterns map. And essentially, it just seems as though in adulthood, there's a shift along the brain axis, but not the behavior axis. And so one way that I've interpreted this is that maybe the relationship between brain and behavior is not as strong in adulthood as what we've observed in adolescence, or perhaps that the brain pattern described in adolescence is also present in adulthood, but the behavioral one is not. Okay. So then, in order to further understand what might be driving a lot of the brain and behavior patterns that we observed, we identified 3 regions of interest in which we chose to perform RNA sequencing of the tissue in collaboration with Dr. Rosemary Baggett, her postdoc Pedrana Cvetkovska, and Pedro Bordignon from the Ledmer Center. And so we chose the anterior cingulate cortex, the dorsal hippocampus, and the ventral hippocampus, and chose to focus on the GD9 time point, as again, this is where we saw the majority of our changes. So very briefly, the way this works is you choose your tissue of interest. So in our case, it was flash frozen tissue from these ROIs. You can then isolate the RNA, generate a cDNA library, and perform your sequencing to get a measure of what genes were being transcribed at the time of collection. And so what we found is that overall, due to the poly-IC exposure at GD9, there were 962 downregulated genes and 668 upregulated genes. Then if we look more closely at the regions of interest, there were 17 downregulated genes in the anterior cingulate cortex, but no significantly upregulated genes. There were 246 significantly downregulated genes in the dorsal hippocampus and 131 upregulated, and 37 downregulated genes in the ventral hippocampus and 12 in the, 12 upregulated genes. And I guess the magnitude of changes that we observe in terms of significant up or down regulation of these genes seems to kind of go along with the magnitude of significance map that we observed in our PLS analyses, which is cool. And another thing I noticed as I was going through this data is that there was one gene that was the most significantly upregulated gene across all 3 regions of interest. So it was 2.2 folds more expressed in the poly-IC group. And this is lipocalin-2 or LCN2 gene, which I hadn't heard about before, but it's conveniently involved in neuroinflammation, which makes sense with the model that we're investigating. It's also implicated in iron transport, which is another component to the inflammatory reaction in the brain, as well as cell viability, movement, invasion, and differentiation. It's also been observed to play a role in inflammation, like via reactive astrocytes, and the majority of work has looked at it in the context of CNS injury. So whether it be stroke or traumatic brain or spinal cord injury, excitotoxic injury, but also neonatal hypoxic ischemia. And in this scenario, it was actually shown that decreasing the expression of this gene also decreased the severity of injury. and other forms of neuroinflammation, so induced by the bacterial mimetic LPS or some sort of autoimmune condition. Okay, so next we took our lists of down and upregulated genes and ordered them by the log fold change, and decided to run a gene ontology analysis using the Panther database to try to understand what some of the gene annotations and processes were associated with the changes we observed. And so here we're looking more for patterns of gene expression based on groups of genes that are either up or down regulated, but this is different than the significantly up or down regulated results that I showed you before. So I know there's a lot going on, but essentially we noticed some key pathways that kept coming up, and this is what I'll highlight. So in the dorsal hippocampus for downregulated genes, we got hits for gonadotropin releasing hormone receptor pathways in yellow, inflammation mediated and chemokine signaling, and cytokine signaling, and then integrin and Wnt signaling pathways as well. And so the latter two are pretty critical to neurodevelopment as well as many cellular functions, but I'll go into some more details in a little bit. In the dorsal hippocampus for upregulated genes, we got, again, the same for pathways as well as some angiogenesis, and it's for a few other pathways as well. And then if we look at the downregulated genes for the anterior cingulate cortex and ventral hippocampus, again, we see the same for pathways being, I guess, the most significant. And for upregulated genes, it just seems to be the Wnt signaling pathway. So just to go over kind of the pathways that kept coming up in a little more detail, we have inflammation mediated by chemokine and cytokine signaling. So this fits really well in the model as we're looking at immune regulation, essentially. So upon binding to G protein coupled receptors, the cytokines and chemokines can control and direct the trafficking and migration of immune cells. If we look at the gonadotropin hormone receptor pathway, these are pretty important in the, I guess, development of reproduction and pubertal development. So these GnRH receptors are located on the pituitary gland, and they can modulate the release of luteinizing hormone or follicle stimulating hormone. And what's interesting about this pathway is it's also very susceptible to stress. Next, if we look at Wnt signaling, this is super critical for the dorsal ventral patterning of the neural tube throughout development. So its expression will determine what part of the tube becomes the telencephalon versus the spinal cord, for example. So it's a very core and highly conserved gene, and a multifunctional protein as well. And then finally, integrin signaling is involved in like a myriad of things, but for example, it's implicated in the migration of neural crest cells, and these are important for the development of your peripheral nervous system, as well as other parts of the body, such as your facial bones and muscles. It's also involved in cellular and neuronal migration, as well as actin reorganization, protein scaffolding, proliferation, the list goes on. So they're all very critical systems to neurodevelopment. Okay, so what I've shown you so far is we've started with this risk factor that's known to disrupt brain development. We then characterize neuroanatomical trajectories due to the exposure, as well as a behavioral phenotype. We've put these together to create kind of a map of brain behavior relationships, and then use this to look further and do some transcriptional profiling. Okay, so next we're going to segue into earlier development. So based on a lot of the hits we got from the Go analysis, looking at many of these kind of basic behavioral mechanisms, not behavioral, developmental mechanisms, and based on the fact that already at P21 we saw differences in trajectory, we wanted to know how early we could detect these alterations in neurodevelopment. So we looked embryonically and neonatally. And this is interesting in light of work from a few years ago showing that serum levels of a pro-inflammatory cytokine, interleukin-6, during pregnancy was actually predictive of functional connectivity networks in babies at 4 weeks of age for both the dorsal attention network in green and the salience network in black. And it was also predictive of executive function in those toddlers at 2 years of age. So this was just an exciting demonstration that maternal immune activation can modulate brain activity at such a young age already. So in order to look at the embryo brain development, we again exposed mothers to MIA or vehicle at GD 9 or 17, and then harvested the embryos at gestational day 18 for ex vivo MRI. And so we collected our samples in 2 batches, achieving between 11 and 17 males or females per condition, coming from at least 4 different litters, again, to account for litter variability. And the scanning was performed in collaboration with Jason Lurch at U of T, who's now at Oxford. And they have a great setup in which you can actually scan 16 samples at a time. And so you can perform an overnight scan of 14 hours and get really beautiful high resolution 40 micron isotropic images. And so here's just a little movie to illustrate the kind of detail that you're able to get from this kind of image. So you can see the whole embryo, we can see the brain, but also many of the organs, which is pretty exciting. And in order to process this data, we used tools developed by Gabriel Devaney, which essentially follow the same theoretical concepts that I described above for the DBM. And so just going into some of the results here, looking at the effects of MIA exposure at GD9 on the embryo brain volume, we see mostly decreases in volume due to this intervention. And if we focus on some regions of interest, we could see an increase in volume in the central subpallidum. And this region is actually what will become the septal nucleus. But other than that, we see decreases in volume in the prethalamus, the hippocampus/ventricular zone. And this is cool because this is kind of a hotbed for neurogenesis in development. So, and this process has been shown to be altered in many neurodevelopmental disorders. So it's possible that we have a decrease here. And finally, we have a decrease in volume in the cerebellum as well. Again, many of these regions have also come up in our longitudinal analysis, which is interesting. If we look at the effects due to GD17 MIA, instead we see completely opposite directionality in changes, with many increases in brain volume kind of throughout the brain. And again, if we focus on some regions of interest, now we see a decrease in the central subpallidum in terms of volume, although it's a slightly different region than what we saw in the GD9. We see an increase in volume in the prethalamus, in the hippocampus subventricular zone. So perhaps now we have an increase in neurogenesis in this scenario, and then an increase in volume in the cerebellum. So I was pretty struck by the difference in pattern due to the 2 gestational timings. Although, of course, it's possible that in this GD17 cohort, we're picking up a difference due to acute neuroinflammation, as the samples were collected only 24 hours after the MIA exposure. And just a quick exploratory analysis, we had the entire body scanned, and given the fact that the MIA exposure is systemic, we thought it would be cool to see whether there were any effects in any other parts of the body. So in the early exposed group, we see a decrease in volume in the liver, for example, or the heart. And in the late exposed group, again, we see decreases in the liver volume, and we also see this prethalamus hotspot show up again. Okay, so moving chronologically, as I mentioned, we also looked at neonatal development, so similar paradigm as before, but now we allowed the offspring to be born and measured behaviors for ultrasonic vocalization at postnatal day 8. So this is a proxy for communicative abilities. We separate the offspring from the mother and measure the number of calls the pup makes for the mother. And after recording this behavior, we prepared the brains for ex vivo MRI.
Lisa, just if you go back 2 slides.
2 slides?
Yeah. Dominique was asking if you saw any changes in the gonad volumes.
I actually haven't focused on that particularly, but that's actually, that would be really interesting to know. I don't know yet, so I'll have to get back to you.
Thanks.
Thanks. Yeah, so this was just to highlight that we have both males and females for all of our groups, again, coming from multiple different litters. And in terms of neuroanatomical changes, we were quite struck to see very few effects due to the MIA timing at this age. So for the GD9 exposed group, we saw very subtle increases in the amygdala nucleus, the right hippocampus, and the right entorhinal cortex. And for the GD17 group, again, we saw very focal and subtle changes in the prelimbic area, the left amygdala, and the crus 1 of the cerebellum, whereas we saw decreases in 2 white matter regions in the corpus callosum and the fimbria, as well as the right subiculum. So again, pretty subtle changes, especially in comparison to what we observed at the other 2 time points. And finally, we looked at the ultrasonic vocalization task by looking at the calls made by every mouse, or every call made by every mouse in the 5-minute recording period instead of looking at summary measures. And we thresholded the calls between 5 milliseconds and 300 milliseconds to eliminate any noise. So since there were no mean differences, we applied the shift function, which essentially allows us to quantify how 2 distributions differ based on the deciles of the distribution, and so how you would have to rearrange one distribution to match another. And so what you could see here is the distribution for the saline group with a median in red. And if we compare that to the other 2 groups, we see that the distribution is shifted for the early poly-IC group with a lower median in call length. And we could then use, and there's no difference for the late poly-IC group. We can then use a percentile bootstrapping technique to determine confidence intervals based on differences in distribution at each decile. And essentially what we see here is that already starting from the 2nd decile of distribution, the early poly-IC group are making fewer calls, and this holds true as the call duration increases as well. Whereas for the late poly-IC group to your right, we see no differences in call duration as the confidence interval crosses the 0 line. And finally, to try to relate the behavioral findings to the brain, we also ran a PLS on the USV and DBM data for the neonates. And what you could see is essentially areas in red are associated with more longer calls. So these are a lot of cortical areas like the auditory cortex, sorry, where's my cursor, the striatum, septal nucleus, hippocampus, and 4th ventricle, whereas the opposite pattern is true for regions of the olfactory tubercle, the thalamus, the amygdala and brainstem. And when we plot the brain behavior correlation, we don't see any differentiation between our groups. So just to tie this all together, I've shown you that there are significant differences in terms of brain development due to both early and late exposure to maternal immune activation. If we look in the embryo brain, we can see that early exposure tends to decrease a lot of the volume of many regions in the brain, whereas late exposure seems to increase that volume. Moving through to the neonatal period, we see we see alterations in communicative behaviors in early exposure, and very subtle increases in brain volume due to late exposure. And finally, if we look longitudinally from childhood to adulthood, we see these interesting deviations in neurodevelopmental trajectory due to early exposure in particular, but that seem to normalize in adulthood. So an interesting next approach would be to determine whether a second hit in adolescence would be sufficient to kind of propel these offspring into a more altered behavioral phenotype through into, or later into adulthood as well. Kind of modeling the two-hit hypothesis. And so with that, I'd like to thank you all so much for your attention, to thank my supervisor, Mallar, as well as members of the Cobra Lab that were super integral to getting this project going and data collection, etc., as well as all our collaborators in Rose's lab and Bridie's lab at the MNI, and Jason at the Mouse Imaging Center. So thank you.
Great, thanks so much. That was a great talk. We have time for questions. I've asked many over the years, so I would urge you to ask them. Sylvia, go ahead.
Yeah, so I was wondering if you add any sex differences. In the second part of the study, it didn't seem like that, but then if you add any, and then related to that, so you didn't show the latent variable that was mainly driven by sex, but I was wondering what that was suggesting, the second latent variable in the first part of the talk, in the PLS analysis.
Right, so I did look at sex differences. It just seems that they're quite subtle. Like there were some regions that seem to have more striking deviations in trajectory in the male, like the septal nucleus. But it's less than I expected. I kind of went into this study really expecting to see some sex differences, but we just didn't pull them out as much as I thought. In terms of the latent variable explaining sex differences, essentially it was just picking up differences in the brain that we know are different due to sex. I apologize, I don't remember off the top of my head what we observed, but in terms of the behavioral findings associated with them, a lot of them kind of were not significant as the confidence intervals actually crossed the 0 line. So it just seemed like we were picking up kind of just basic differences in brain due to sex. Yeah.
Thanks.
Thank you.
Veronique, go ahead.
Yes, hi, Elisa. This is Veronique Bogot. A fascinating talk. I loved it.
Thank you.
I was wondering whether you thought that whether behavioral experiences could actually modulate some of the phenotypes down the line. And I'll tell you why. I actually have a project that I did with Jason Lurch a while back, which we published in 2011, where we showed that if you train rodents, mice, we did this in Toronto, on a task that stimulates the hippocampus, then it will have an impact on gray matter in the hippocampus. So if you train them on spatial memory, you're going to grow the hippocampus. If you train the mice on a stimulus response strategy task, it's like a version of the water maze where you put a pole and then the mice just learn to swim to the pole. Mm-hmm. Then we actually increased gray matter in the in the striatum. And then in another study that I did with Jude Poirier here at the Douglas, we found that there is a relationship between these strategies and cytokines. And I can't wrap my head around, you know, like what causes what and whether we can reverse the changes and modulate the changes with behavioral. So we're trying to run these experiments, but I was really wondering what you thought, because this could be a factor that could be really important in the modulation of phenotypes that lead to psychiatric disorders. And another reason why I'm saying that is that if you give young adults video games that stimulate the striatum, you get a shrinkage in the hippocampus, which we know is a risk factor for schizophrenia, for example. And I've published about 5 papers on schizophrenia showing they have an impairment in spatial memory, etc. So I don't know. I loved your talk, and I was just wondering what you thought about the interaction between experience-dependent modulators and cytokines and development, and then that impact on phenotype.
Oh, thank you. Yeah, I mean, it's kind of hard to disentangle, I suppose, because we're like, I find that anything that you do to these mice is going to affect them in some way, right? But I guess one way that some others have tried to separate these two is they'll perform only imaging in some mice and only behavior in others. But then the challenge there is that it's difficult to then make any conclusions about the two together. So yeah, it's definitely a difficult question to try to address.
Okay, well, I think you're in a great position because you can actually, you can manipulate these variables. Mm-hmm. Whereas with humans, it's a little harder. We just observe
Definitely.
them.
Definitely.
Except with video games, the only time I was able to successfully shrink their hippocampus, showing a causal, it was a longitudinal study. But anyway, so I think, yeah, great work.
Thank you.
Dominique, go ahead. You'll need to unmute yourself.
I should, yes. Thank you. Thank you very much, Elisa. This is a great talk and a lot of very nice data. I really enjoyed it a lot. I have 2 questions for you. Oh, maybe 1 question and 1 comment, if that you can comment on. You showed that you have changes in the volume in the subventricular zone at some point, and I can't remember quite now in which condition, but my question was, have you looked at some neurogenesis in the hippocampus, for instance? Do you know if those changes relate to or lead to changes in neurogenesis in the hippocampus later on?
I haven't looked at it. I would really love to so that was in the embryos that we saw those effects, and we have all those brains. So I was waiting to have my final MRI data before diving into the histology, but that's something I'm really interested in seeing, is whether we actually have differences in neurogenesis or whether that volume difference is due to something else. I know a lot of work has been done in the MIA model. I believe, though, the MIA was at GD 12.5, which I guess corresponds with more of the peak of neurogenesis. And with that paradigm, they did see, I think, increases in neurogenesis in that case. So yeah, I'd be really curious to see, A, are we affecting it, and B, is it different based on the gestational timings? But I haven't done it yet. So
and maybe 1 comment and a question, if you want. You were talking about the second hit that you would be interested to look at, and I mean, if I were to design this type of experiment, I would compare an immune challenge with something totally unrelated, like another stressful challenge, because really, I mean, animals might be more prone to react differently in terms of the immune activation, but they might also have a generalized sensitization that you could maybe unravel using a different type of stimulus. So I think it would be nice to compare something that is really unrelated to any type of cytokine activation.
Yeah, we're actually quite interested in looking at adolescent THC exposure. As it's been highlighted as like a pretty significant risk factor for psychosis. So that is the project Mallar mentioned that we were starting just before COVID. So we'll see how that comes together. But certainly other types of second hits would be super interesting, like, any kind of stress paradigm I know has been looked at in relationship with MIA. And but yeah,
good. You have lots of work ahead of you.
Yeah, seems like it.
Yeah,
Thank you, Dominique.
So what a good thesis should do, it should open up all sorts of other investigations that you can keep doing for postdocs or some sort of career, right? We have 2 more questions, and then I'm going to probably cut this off. So Dan, go ahead.
Hi, can you hear me now?
Yeah.
Okay, cool. I was just wondering, so when you presented your longitudinal data from the same mouse scanning over multiple periods, the general pattern was that the early treatments would overshoot and then come back to normal by adulthood, right? Yeah. Did you see, were there certain brain regions of the ones that were significant, were there some that made it back to normal and some that still in adulthood were deformed, I guess you'd call it? Or did they all reach back to normal? Does that make sense?
Yeah, I want to say that most of them did more or less normalize. Off the top of my head, like that trajectory that I showed you was the one that seemed to come up more often. I'd have to revisit the data to double check because it's been a while since I've done those analyses, but overall the pattern seemed to really be in normalization. So we were commenting with some of my colleagues that if I had chosen just to image them at that postnatal day 90 time point, we would have seen no differences basically, which is interesting.
Really? Okay. I'd be really interested, because I'm interested in adolescent brain development. I'd be interested to know, when you publish this, or if you are looking at the data and you happen to come across that information, if you're interested in sending it to me, be curious to know if there's a relationship between how back to normal they're able to get and how much development we know there is in adolescence in these brain regions.
Mm-hmm.
So,
yeah.
Anyways, that's just a quick question. Thank you very much. It was a great talk.
Thank you. Thanks.
And Masha?
Hi, Elisa.
Hi.
I just have a question. I was wondering, do you know how maternal immune activation is measured in pregnant women?
In humans usually?
Yeah, in humans, yeah.
Yeah, so I think there's a variety of different ways. Like a lot of the original epidemiological studies actually just took, these are the years that we knew there was a viral pandemic. These are all the moms that were pregnant. And then this is the likelihood of offspring developing psychosis. And then after that, what they do is they measure, you can either measure cytokine levels in the blood, or you can measure like antibody levels. So that would be for something like Toxoplasma gondii or other types of infections. So it kind of depends on the infection, but you're essentially taking a blood sample from the mother at different gestational time points.
Okay, awesome. Thank you so much for answering that question. And thanks for your talk. I really enjoyed it.
Thank you.
That's a good place to, we're already a little late, so it's a good place to maybe stop. So I'm going to unmute everybody so we can give you the appropriate amount of applause for such a wonderful talk. So thank you very much, Lisa. Great work.
Thank you all.
See you all later, folks. See you next week.
Bye.
Thank you.